On to the very important part of this meeting, and all of us have to be keyed on. The keynote lecture of this webinar is to be delivered by Dr. Gordon Plant, who is going to be talking on understanding and unlocking the potential of OCT in neuro-ophthalmology. So we all look forward to hearing some amazing inputs from you, Doctor. On to you. Could you share your screen?
Right. Can you, am I sharing my screen?
Yes,
Can you hear me?
Yes.
Excellent.
Yes.
Started. Okay. Well, I feel very privileged to have 15 minutes for my talk. You've all done remarkably well. It's been a wonderful, wonderful session. And what I thought I'd do is I first came across OCT 30 years ago, and there have been certain points at which I've seen where it really is the way forward. And I thought I would take you through those steps. So I think OCT has been as important to ophthalmology as the invention of the ophthalmoscope nearly 2 centuries ago. And my hospital, the Neurology Hospital in Queen Square, was opened in 1860. So the ophthalmoscope was new technology. And the yellow arrows there point to 2 ophthalmologists. The one on the left is Marcus Gunn, who you have all heard of. The other is Brudenell Carter, who in fact was a pioneer of optic nerve sheath fenestration back there in the 1880s. The 2 red arrows, one is Hewlings Jackson and the other William Gowers. And they both worked at Moorfields and were experts with the ophthalmoscope, in some ways more so than the ophthalmologists. Gowers wrote a famous textbook of medical ophthalmoscopy. So there was fusion of neurology and ophthalmology, and here's what happens when it goes wrong. Here is a whole mount of a primate retina with the projection of the central retina stained with horseradish peroxidase. And can you see my pointer?
Yes, we can see.
Good, yes. And you can see that this, which has been called the papillomacular bundle, is not just the projection of the fovea, but also of all the ganglion cells between the fovea and the optic disc. But it was so firmly held that this was the projection, the papillomacular bundle was the projection of the fovea, that people actually invented axons which went at right angles to the other axons, and even invented a field defect corresponding to their damage in glaucoma. So it's very important that we keep neurology and ophthalmology together. So what is the relevance of Michelson? Well, it was Michelson's interferometer that's set up, which he developed to measure the speed of light, which was used in the first OCT machines. His equipment was 22.5 miles long. So it doesn't immediately sound very promising for ophthalmology, but here we have James Fujimoto, and that first time I ever saw an OCT back in 1991, there it was. It didn't look very promising, did it? But here we are now with all of these developments. So the 3 things that I've been involved with are the residual deficit in optic neuritis, transsynaptic degeneration, and intraretinal changes in multiple sclerosis. So we've been hearing about optic neuritis, and one of the debates that I grew up with was whether the primary deficit in MS is due to demyelination or due to axonal loss. here's an entire optic nerve from the back of the eye to the chiasm, stained for myelin in a patient who died with multiple sclerosis. And vision wasn't too bad in life. And you can see that extensive demyelination. So Bill Hoyt was a pioneer in this respect. People traveled to his lab, to his clinic, to learn how to look at the nerve fiber layer using the direct ophthalmoscope. And back in 1974, he published these images showing axonal loss in the retinal nerve fiber layer in multiple sclerosis. He could not get this published in Neurology journal because they said, oh, this is not relevant. MS is a demyelinating disease. Axonal loss is not important. Well, we'd looked at remyelination by following up VEPs in patients with optic neuritis over some years. And although there was improvement in the VEP delays suggesting remyelination, this did not seem to relate to any improvement in vision. And we'd also shown that in acute optic neuritis, neurofilaments appear in plasma, which are markers of axonal damage. So the OCT by this century had reached a point where we could start to use this to look at axonal loss after optic neuritis. And this actually is now my most cited paper. And I think the reason that it was successful was because I didn't look at a random selection or an uncontrolled selection of patients with optic neuritis. I actually selected patients with a range of visual loss from minimal to the most severe, which of course after MS optic neuritis is relatively unusual. And by doing this, we were able to show that the visual deficit correlated very much with axonal loss and not with the delayed VEP, which is the only marker we have really for the extent of demyelination, something we couldn't have done without OCT. The other thing that Bill Hoyt taught me when I was in his clinic in 1991 was to look for nerve fiber loss in the retina in patients with congenital hemianopia. And he didn't believe that you could see nerve fiber loss in patients with acquired hemianopia. Hey, could you mute, please, if you're not speaking? Thank you. When I was a medical student, I did some studies on with Hisako Ikeda on kittens reared with convergent squint, actually a paralytic squint. She used to remove the lateral rectus muscle to produce this. And we showed that there were changes in the lateral geniculate nucleus in patients in these kittens with paralytic strabismus. Before then, it had only been shown due to occlusion. And so I've always been very keen on taking advantage of the layout of the visual system with its single synapse between retinal ganglion cell and visual cortex. And these studies began to appear in glaucoma patients saying, oh, they've got brain disease. They've got atrophy in not just in the lateral geniculate, but also in the visual cortex. This led me to wonder whether these changes were secondary or primary, and also whether other changes which were seen in the retina in Alzheimer's and Parkinson's were secondary to the brain disease or due to primary involvement of the retina. And you're all familiar with Wallerian degeneration. Here's Augustus Waller, who used this as a way of tracing fiber pathways in the CNS. You cut an axon and you follow the degenerated distal part of the axon. That's Wallerian degeneration. But we can see neuronal degeneration following damage to a neuron, not only Wallerian, which is anterograde, but also retrograde to the cell body. and also both retrograde and anterograde transsynaptic. We've reviewed the possibility that all of these changes, which are seen in the central nervous system in glaucoma, can be explained by damage at the level of the retinal ganglion cells, and these are the various locations and how we can image this type of damage, but it can all be ascribed to retinal ganglion cell damage. And I was also aware of this interesting study from the 60s by Van Buren, who had sectioned the chiasm in primates and looked at the retina 20 months later. And as expected, this is retrograde degeneration. There were no retinal ganglion cells on the nasal hemiretina, but also this cystic change in the internuclear layer. We'll come back to that later. But he also carried out occipital lobectomy and looked at the retina at 48 months. And there are still surviving ganglion cells, but much thinned, corresponding to the hemiretina involved with the loss of vision. So this was against what Bill Hoyt had taught me. And we also had a classic example of an N of 1 study. Neil Miller and Steve Newman, they found the oldest person they could with the longest standing occipital stroke, and they couldn't see any changes in the retina using these techniques that Bill Hoyt had taught them to look for nerve fiber loss. We all know that we saw an example of band atrophy earlier, and we all know that it's difficult to see. It's difficult to persuade people that it's present in cases like this. This is as severe as it can get. This is a chiasmal transection in a human. And we also know that we see it with tract lesions, because this is again direct retrograde degeneration. And Bill Hoyt produced this lovely figure explaining the pattern of loss with a homeopic loss in one eye and band atrophy in the other. And I find it easier to explain band atrophy to people in cases like this, which is so-called twin peaks papilloedema, because here is the band atrophy. There are no nerve fibers here, so we can only get papilloedema at the upper and lower folds. Those of you who speak Spanish will know that the twin peaks in San Francisco were named by the Spanish after certain features of female anatomy. And we went on to show triple peaks papilloedema in this patient, but that's something that your ancient civilization came up with many years ago. So here's a retro-chiasmal lesion. Can anyone see the band atrophy in one eye and the mild subtle atrophy in the other eye? So initially we took cases with very long-standing lesions, possibly congenital, and showed that with OCT, very difficult clinically, but this type of band atrophy that you see with chiasmal lesions, you can see in the eye with the temporal hemianopia, can detect this very easily on OCT. So there's a right hemianopia, here's a left hemianopia with band atrophy. And we then looked at a much larger group, and these are the eyes with nasal hemianopia, and we had an acquired group, a congenital group, and a control group, and there was the greatest thinning in the congenital group, fitting with the fact that it's visible with ophthalmoscopy, but there was also thinning in the acquired group. This is how it looks in the eye with the crossing fiber defect, but you can see this much more clearly if you take the sectors, here they are, and look at them in a mirror symmetric fashion as ratios. So what you get now is for the controls, you get a ratio of 1 because there's no difference between the two eyes. And now you can see where we have greater loss of non-crossing fibers and greater loss of crossing fibers. So this is now more detail in the familiar band atrophy. You can see that it's really is the nasal part of the disc that has the greatest proportion of crossing fibers. Now, of course, along has come, I mean, we had to jump through hoops to prove it, but now we can look at the ganglion cell layer, and it's like looking at a visual field. There's a congenital hemianopia, and here we have hemianopic sectors, partial quadrantic lesions. Actually, we did publish a paper on homonymous quadrantinopia, but it was very difficult to show convincingly with the nerve fiber layer. We needed a lot of cases. Here now you only need one. So, and we were also able to show by looking at two groups. This was a group of 38 patients with long-standing hemianopia, and we were able to show how the thinning occurred very, very slowly over a matter of years. Here's the age of the patient, the duration of the occipital lesion in log years, and this is the relationship with this surface. So it's a slow loss that gets slower as time goes by. And then here are a few cases we looked over some years following their stroke. And other than this patient who probably had, who did have lateral geniculate nucleus involvement with the stroke, you can see this is a slow process carrying on over a matter of years, but it really does occur. So it's interesting to look again at the congenital cases to see what kind of residual vision they have compared to these acquired strokes. And of course, there are many attempts at neuroprotection, but that tends to be for the damaged axon. This is a much slower process, and I think holds much more hope for neuroprotection because of that. So, and very little is known about the what actually causes this. So this is the retina in MS, and Ari Green came up with this very interesting paper on pathology of MS retina. And here's the normal retina with the inner nuclear layer here. And you can see this cystic change in the inner nuclear layer of the MS patients. And he went on to show that this can be demonstrated on OCT, as you can see here, and then it relates to the severity of the disease. So this interested me again because I had seen this. If you cut the chiasm, Van Buren showed, you get this massive cystic change in the inner nuclear layer. And this has also been reported in human pathology, pathological studies of the eye in patients who in life had optic neuropathy. So what we did was look at these. I've been studying this epidemic, now endemic optic neuropathy in Tanzania since the 90s. And we managed to get an OCT machine out there. And they have this very characteristic loss of this centrocecal projection that I was talking about earlier. And we were able to show that they also get this cystic change. And here it shows very nicely on the infrared. And here is the cystic change here. And in fact, it relates entirely to the nerve fiber loss. So this is much more like what Van Buren was showing on a lesser scale. And the more severe the axonal loss in these patients, the more likely you are to see this cystic change. So, it's had an unfortunate episode in terms of nomenclature, because there is already something called microcystic macular edema in the literature, but that's what they wanted to call it. But Abegg did something different from us. He just looked at a whole range of optic neuropathies from any cause at all and found this change. So he proposed that this is a retrograde maculopathy, which certainly is what we suspected in our cases, so a transsynaptic retrograde maculopathy. But we know that inflammation does occur in the retina in MS. So it may be that there's two things going on here. There is inflammation in the retina causing this edema, but there is also a more permanent change due to axonal loss and retrograde degeneration. So this is when I was registrar at Queen Square. Ian MacDonald published this review of fluorescent angiography and acute optic neuritis. And in fact, these changes are not related directly to the optic neuritis. It was just as likely to see them in the other eye. So what we're doing now is combining OCT with neurophysiology. And we have a paper in submission now, which shows that there is a relationship between the P50 amplitude of the pattern EMG, which reflects retinal function, particularly bipolar cell function, as opposed to the N95, which is ganglion cell, and that relates very clearly to nerve fiber loss and ganglion cell loss. But we find there is a correlation between the inner nuclear layer thickness and the P50 amplitude. And so what we now don't know is whether this is a transsynaptic change, such as seen with any optic neuropathy, or whether this represents specific inflammation in the retina in multiple sclerosis. So that's been my relationship with OCT over the last 30 years since I first saw the first image. But please take this as an indication that your group here is absolutely fantastic. You're all well entrenched in both neurology and ophthalmology, and that's the way we need to be. But we need to educate our colleagues as well. We also need to make sure we continue training neuro-ophthalmologists and medical ophthalmologists. And thank you very much for asking me to speak and to attend this wonderful meeting. And thank you again. That's the end.
Thank you very much. That was an amazing talk we all heard. Could you stop sharing your screen?
There we go.
Thank you. Murli, do you have anything to ask before I give my concluding remarks?
Yes, Dr. Gordon, is there a difference in the pattern of nerve fiber loss between neuromyelitis, NMOSD, neuromyelitis optica spectrum disorders, and optic neuritis and MS when you study them on OCT? Did you find any difference?
Much worse, much more severe. That's the main difference. And of course, there is less evidence of demyelination with the NMO. There probably is some demyelination, but NMO seems to be predominantly a glial cell immune condition, and is much more, causes much more axonal loss. So that's the main difference. You get the microcystic change in the inner nuclear layer. You get that with all the different types of optic neuritis.
So I think it's been a long day. So I should conclude. A wonderful, wonderful webinar it's been. And I don't know about each of you, I really learned so much today. This could not have been possible at all without the expertise of this great panel here. And of course, the very great talks and so crisp and succinct talks of each and every one of you. My thanks are due to my co-moderator, Dr. Murli, who I'm sure all of you would have realized that he has just left no stones unturned to make it a truly remembered webinar. My thanks are always due to AIOS admin, Mr. Kripal, and his team, our very able Mr. Sunil, who has been the webinar admin and is always very committed and sincere to ensure that the webinar goes off sequentially. My thanks to Sai and Manjula from Numerotech for their background support at all times. My most heartfelt thanks to Entold for helping me realize this dream to reach out to one and all of you in this audience, because they have sponsored every event of ours. And finally, and most importantly, my thanks to our dear attendees for attending in full strength to make it such a great webinar. Thank you very much. Thank you, one and all of you. Thanks a lot.
Thank you.